Good morning, everyone. My name is Hanny Rivera. I'm a postdoctoral investigator in Boston University in the Davies Marine Population Genomics Lab. And today I'll be talking to you about gene expression and physiological changes under thermal stress, heterotrophy, and symbiosis, a symbiotic state in the facultatively symbiotic coral Oculina arbuscula. Now, endosymbioses are really fascinating because they're found across the tree of life. And by endosymbiosis, I mean processes and relationships through which another organism lives inside the tissue of a different organism in a mutually beneficial relationship. So here, just to showcase a few examples, are salamanders that have symbiosis with algal cells in the egg stage. We, of course, have lichens that are this tripartite symbiosis, and things like legumes and rhizobium that help plants fix nutrients that they cannot otherwise get, and the endosymbiosis that I'll be talking to you about today, coral algal symbiosis, where endosymbiotic dinoflagellate lives inside the coral animal tissue and provides it with photosynthates from photosynthesis. Now, endosymbionts really integrate with the cell to maximize mutual benefit. This is a diagram from a review of coral algal symbiosis published a while back. And in it, you can see that the symbiont cell lives inside the coral host cell in this specialized sort of habitat called the symbiosome, which is really an arrested phagosome. And within that environment, the pH of the environment is very tightly regulated by the coral host, as is the cycling of all sorts of nutrients that the symbiont needs in order to photosynthesize effectively. And in exchange, the symbiont translocates a lot of lipids and sugar molecules to the host as sort of payment for its nice little home. Now, one thing that's interesting about this symbiosis, and it happens in a couple of different systems, especially within Cnidaria, is that we see a somewhat of a suppression of the immune system when the symbiosis is established. So here on the right, I'm showing you fluorescence microscopy images of staining that is specific to NF-kappa B, which is a master immune system regulator. And now these are cells of the anemone aptasia that can both live happily without symbionts or can uptake symbionts as larvae. And you'll notice that when they are aposymbiotic, when they have not been given any symbionts to uptake, their NF-kappa B expression is pretty widespread. They have very consistent NF-kappa B expression across the entire larval body. But once they are infected with the symbiotic cells, that NF-kappa B expression pretty much shuts down almost entirely. And so this I find to be a very interesting pattern because it suggests that there's some kind of pretty intense trade-off here where having this symbiosis can somewhat leave you in an immunocompromised state. And so some of the questions that I seek to explore in today's talk and in some of the experiments that I've conducted at the Davies Lab is to better understand how symbiosis in corals might lead to changes in immune response genes, how conserved these responses are across other taxa that have similar symbiotic, endosymbiotic partners. Is it always the case that there's immunosuppression in these very tight-knit endosymbiotic relationships? And then I also want to look a little bit at some of the other physiological trade-offs that might be associated with symbiosis, especially combined with other stressors that are very common and very pertinent to corals in particular, such as heat and feeding or food limitation. So the organism that I'm going to be using to answer these questions is known as Oculina arbuscula. It's a calcifying coral, but it's not really like a major reef builder. It only makes really fairly small colonies, but it does secrete calcium carbonate. The range of this coral is mostly shown here on the right-hand side in this map, and it extends from about North Carolina down to about midway through Florida. You can find it a little bit further south, but it becomes much more rare there. It hosts symbionts in the genus Braviolum. You can see those up close here in panel A under the microscope. And what makes this coral really interesting is that unlike its tropical counterparts, the normal Caribbean reef-building corals, it can live in an aposymbiotic state, in a quote-unquote bleach state that you might have heard, without really dying or being in a very stressful state. So because it's found in this sort of subtemperate region, it tends to be a very active feeder and can therefore survive without symbionts for prolonged periods of time without any sort of physical manifestations of stress. And we can also manipulate the symbiosis in the lab by exposing it to menthol. So here on the bottom are two fragments of the same colony, one of which I exposed to menthol and essentially caused complete expulsion of the symbionts, and the other which has maintained its symbiosis. And so this gives us a really powerful model system in which to control for the coral host genotype while manipulating the presence or absence of the symbiosis without causing some severe stress in the organism. Okay, so in order to answer some of these questions, I'm going to present a couple of these analyses to you today. So in terms of thinking about changes in immune response genes, I'll be presenting some RNA-seq of aposymbiotic and symbiotic colonies. In order to compare how conserved these responses are across different taxa, I've looked at gene expression data sets, the one that I created for Oculina, as well as two other available ones for Aptasia, and that's salamander and bistoma, and done a cog analysis to see how similar the responses are at a very sort of meta level of function. And then in terms of physiological trade-offs, I'll be talking about NF-kappa B levels of Oculina arbuscula under heat stress and feeding stress, as well as some other physiological changes in carbohydrate and protein levels. So for RNA-seq, we took 5 different colonies that had both symbiotic and aposymbiotic fragments, and we sampled all of those in a paired design. Transcriptome and assembly code are all publicly available on Davies Lab website. And so just to give you a very quick overview, gene expression is strongly influenced by symbiotic state. So if we look at the influence of symbiotic state in both GENET, they're both significant drivers of gene expression across colonies. And then within the differentially expressed by symbiotic state, we see a lot of enrichment for gene ontology terms that are associated with immune processes. So there's a lot of defense regulation and immune system processes, and there is even downregulation of NF-kappa B signaling, which, harkening back to those results from Mansfield et al. About Aptasia, really highlights that this seems to be a somewhat conserved mechanism. And indeed, when I actually look across different taxa using COG, terms to compare orthologous gene and functions across very divergent taxa, I see that, again, defense mechanisms at this very high level overview is consistently downregulated, and very significantly so, in both Oculina, Aptasia, and Apistoma, which is actually a vertebrate salamander. And it's actually the most kind of consistent category across all of these COG terms. So that seems to be really interesting in terms of highlighting this as a potential trade-off between having a very close-knit endosymbiosis that may be very beneficial but may actually come with some immunocompromisation. So one of the things that we then looked at was heat stress and how that regulates NF-kappa B. So for this we did a stress experiment with only symbiotic colonies and put them under controller heated conditions of temperature, and then controller fed conditions, unfed or fed conditions as well, so a full factorial. We didn't get NF-kappa B protein levels for 2 of the treatments, so here I'm just showing you unfed controls versus fed heated colonies, but what we see is that under heat stress, even when you are fed, you still upregulate NF-kappa B quite a bit. So what this sort of to relate this to the immune regulation part of the symbiotic state, right, is if NF-kappa B is an important response in heat stress, and having certain types of symbionts might limit your NF-kappa B expression levels, it could therefore then compromise your response to heat stress. So we also compared carbohydrates and protein levels in both the host and the symbiont across these feeding and heat stress treatments. And we find that for the host, carbohydrate levels really do not change across all 4 treatments. In the symbionts, we see a slightly lower level of carbohydrates when they are fed and heated. So this might be that within the coral host, right, when there is higher feeding, not only is there bleaching, but there might be sort of lower translocation of photosynthates from the symbionts to the host, because they might not need it as much from those symbionts. In terms of protein levels, we see a very different response. Proteins are much more sensitive to heat stress, at least in this experiment, and we see that for the coral host, levels are much lower in all of the sort of control conditions. So even when they're unfed, we see a decline in protein levels, and definitely under heated conditions, both fed and unfed. For the symbiont, we see a similar response, except that in this case, the unfed heated treatment was not significantly different from the fed control, just because some of these points up here, some of the samples were much higher in their protein levels. So I mentioned earlier that one of the connections between NF-kappa B and this sort of heat stress response might come in with different types of symbionts. And so there are many different genera of symbionts and dinoflagellates that associate with cnidarians, one of them being Symbiodinium and Durastinium. Now, Durastinium tends to be a much more thermally tolerant genera. So corals and anemones that host Durastinium tend to bleach less during high levels of heat stress, whereas those that host other clades, such as Symbiodinium or Cladocopium, will bleach much more easily. Now, one thing that's really interesting is that at least in Aiptasia, we see that when they uptake Symbiodinium, there is that suppression of NF-kappa B pretty widespread throughout the entire larval body, but that suppression does not actually happen when they are hosting Durastinium, that more thermally tolerant genera. And so this begs a question in my mind as to whether the thermal tolerance that we see when cnidarians are hosting Durastinium is really a result of the symbiont itself being more thermally tolerant, and there's some evidence to support that. But there may also be an interactive effect of this immunosuppression that happens when they're hosting something else. Whereas hosting Durastinium may actually allow their immune system to still be sort of ready to respond to heat stress through other pathways. Whereas when they have associations with other symbionts, that response might be more limited because there is such widespread suppression of NF-kappa B, which is this master immune system regulator. Further. But just to quickly summarize some of our findings, symbiosis causes a lower expression of immune-associated genes. This pattern is conserved across taxa. In corals, lower NF-kappa B expression associated with some symbiosis may also limit their ability to respond to heat stress. And so some of the next steps that I'm taking is looking at NF-kappa B levels in aposymbiotic and symbiotic fragments of Oculina or arbuscula to see whether those vary based on symbiotic state. I'll also try to conduct some experiments to determine if symbiotic corals have more compromised responses to immune challenges such as pathogenic bacteria. And I also want to investigate the potential mechanisms of immunosuppression by symbionts by looking at single-cell RNA-seq of aposymbiotic and symbiotic fragments. And with that, I'll just quickly acknowledge those members of the Davies Lab that had helped make this work possible and have helped, kind of fine-tune these presentations and papers that are associated. Also like to thank Hui Computing for some of the bioinformatic capabilities, as well as sample collection by Steve Broadhurst down at UNC.